My name is Prisca Obi, and I'm a PhD student working with Virtus Therapeutics, a Yale University spinout that's pioneering new small molecule treatments for orphan metabolic diseases. This is a picture of the Gravina family. In 2015, they welcomed twins into the world, a boy named Isaac and a girl named Charlize. Both twins were diagnosed with propionic acidemia, a rare genetic condition affecting 1 in 100,000 babies. Due to this disease, they're missing an enzyme that their body needs in order to digest proteins and fats in their diets. And less than a week out of the hospital, both twins underwent acute metabolic failure. Unfortunately, there is no cure or targeted treatment for propionic acidemia, only ways to manage the symptoms. So Isaac and Charlize were placed on a special synthetic diet that was supplied by feeding tubes that were inserted into their stomachs through their noses. And when this wasn't enough to keep their symptoms under control, the doctors referred them to the only other option, a liver transplant. Here's a picture of the family 4 years later. If you'll notice, Isaac is absent from this photo because he sadly passed away after complications from the transplantation. The parents made a difficult decision to still have his twin sister Charlize get a transplant. And as you can also tell from these photos, she fortunately survived, but only after receiving 2 transplants due to the first liver failing. Unfortunately, this is the unsatisfying reality of several patients with propionic acidemia. And even for those who survive early childhood, they're at risk for several long-term complications, including intellectual disability, seizures, heart problems, vision loss, and kidney disease, all adding up to an increased risk of dying by young adulthood. Our goal at Virtus Therapeutics is to develop the first FDA-approved small molecule therapy for propionic acidemia, based on our newly discovered drugs called VTACs, or Virtus Therapeutics Activator Compounds, that are currently under strong IP protection until 2040. VTACs consist of a growing family of 14 small molecules that were discovered at Yale University for their ability to activate an enzyme known as PANK3, increasing its selectivity by nearly 200% compared to control. PANK3 is a critical enzyme because it produces coenzyme A, which you can think of as an essential vitamin our cells need in order to carry out basic functions, including producing energy, making neurotransmitters, expressing genes, etc. In propionic acidemia, toxic metabolites accumulate in the body that have been shown to inhibit PANK3. This leads to a deficiency of coenzyme A and metabolic failure. Our VTACs aim to reverse this by boosting the activity of the PANK3 enzyme, which leads to restoration of coenzyme A levels and ultimately metabolic rescue. For patients, VTACs would mean fewer life-threatening metabolic crises and hospitalizations, less chronic complications, an increased span of life, and better overall quality of life. Currently, there is no other small molecule treatment in active clinical trials for propionic acidemia. Our main competitor is an mRNA enzyme replacement therapy that's presently in phase 1 for this disease. VTACs will be formulated as an oral pill or liquid medication that can be taken once a week. As such, it will be safer, more convenient, and cheaper than mRNA therapy, which will require patients to visit the hospital 1 to 2 times a month for IV infusions of the mRNA, which could have unknown long-term side effects. We estimate that the market size for our drugs will be $350 million a year, based on there being about 1,000 patients with this disease and pricing on par with similar drugs. We expect this market to grow as patients survive longer from better treatment and anticipate being able to expand it into international markets, especially in countries where this disease is more prevalent. Lastly, there are over 20 other orphan metabolic diseases that are also marked by coenzyme A deficiency, meaning that VTACs have a high potential to be a multi-indication drug with an even larger patient market. To fund these efforts, we have raised nearly $2 million of non-dilutive funding from both public and private sources and are currently being funded by a phase 1 SBIR that was awarded to Virtus Therapeutics. This money has gone towards basic research that led to VTAC discovery. We've opened up a physical lab site in Groton, Connecticut, where we've hired a scientific director and have begun preclinical studies. Our next funding goal is to secure a seed round investment of $7 million, which will enable us to do 3 things. 1, complete our preclinical studies that show the efficacy of our drugs in mice. 2, perform IND-enabling studies to test the safety of VTACs in a non-rodent model. And 3, hire a CEO and scientists to carry this out over the next 4 years with the ultimate goal of getting to first-in-human studies. This is part of our long-term drug development plan in which we aim to start expedited clinical trials in 2028 with the help of a strategic partner, followed by acquisition by pharma to help carry out phase 3 trials. We predict that VTAX can get FDA approved as soon as 2036, and upon market adoption will receive 12 years of FDA protection against generics due to their pediatric and orphan status. To end this, I would like to acknowledge the current team at Virtus that makes this all possible. VTACs were discovered by Dr. Chirpuri Mamoon, the company founder, who is also a professor at Yale, who has been studying metabolic pathways and their relevance to human health for almost 20 years. Together, our team has been awarded over $10 million in NIH research funding, published over 100 scientific papers, garnered over 30 years of commercial experience in the life sciences, and have been involved in the development of 2 clinically successful drugs. We hope that you are able to join us in our efforts to move Virtus Therapeutics forward. Thank you.